Dear students, welcome back to the next part of microscopy. Today we will be discussing about fluorescent microscopy and mainly the electron microscopes that are most commonly used. So now let us start with fluorescent microscopy. Okay. So what is a fluorescent microscope? It exposes specimen to ultraviolet or blue light. Okay. So specimens are usually stained with fluorochromes. Now these fluorochromes are nothing but dyes, fluorescent dyes that are being used for the fluorescent microscopy. It shows a bright image of the object resulting from the fluorescent light emitted by the specimen. So who will emit the, means from where the light will be emitted? So it will be emitted by the specimen. The fluorescent light will be emitted by the specimen and that will be shown as a bright image in the microscope. Okay. So basically the microscope looks like this, and when the image is produced it will form this kind of fluorescent images. This kind of fluorescent images on a dark background. Okay, with a dark background they will produce this kind of fluorescent images. Now let us discuss about the principle of the fluorescent microscope now. So certain dyes called as fluorochrome, after absorbing UV rays, gets raised to a higher energy level. So certain dyes are there. So certain dyes are there which are called as fluorochromes. After absorbing UV rays, they get raised to a high energy state or high energy level. So when the dye molecules return to their normal state, so as when they reach into an excited state, thereafter they will again return back to the normal state. So when they will return back to the normal state, what happens is that they releases excess energy. Okay. So this excess energy will be released in the form of visible light, and that will be observed as a visible intensity in the microscope. Okay. So this will be produced in the form of a fluorescent image. Now if you see this diagram which will depict the principle of this particular microscope. Now here you can see that this is the specimen. Specimen is here. Okay, just above this. So the specimen is here itself. Here itself, the specimen is here. Now the fluorochrome dye the fluorochrome dye is already being attached with the specimen. So now they will emit certain fluorescent lights. Okay. Now what will happen? This is the illumination source of the microscope. Now this illumination source, from this illumination source, the wavelength or the light rays will fall on this excitation filter. Now what will happen next is this excitation filter will excite the lights. Okay. Now this light will be passed through them and they will reach a mirror called dichromic mirror. Okay. Now what is the role of this dichromic mirror? This dichromic mirror, what it will do, it will reflect certain kind of excited lights towards the specimen. Okay. Now specimen is here and there is a objective lens as well. Now what will happen? They will reflect this light towards the objective, and on the same simultaneously what will happen, from this particular molecule the fluorescent light will be emitted, and this particular dichromic mirror will allow this fluorescent light to go towards the detector. Okay. And for that reason what will happen, this particular image will be converted into a fluorescent image which will be detected finally by the microscope, and then we will be able to visualize them in the form of a fluorescent image. So next we will be discussing about electron microscope. So this is the last part of the microscopy. Electron microscopes were first co-invented by Max Knoll and Ernst Ruska, and it was done in the year 1931. Okay. So it was done in the year 1931, co-invented by two persons. One was Max Knoll and the other one was Ernst Ruska. Then electron microscopes uses a beam of highly energetic electrons. As I have already told you in the first class itself, that these electron microscopes never uses light as an illumination source. Okay, mind it. And remember this. Here, electron microscopes, what they does, they uses a beam of highly energetic, okay, highly energetic electrons to examine objects on a very fine scale. So when you will see an image with the help of an electron microscope, it will provide you a very fine quality image, okay, and that too of very minute structures or very minute organisms like virus even. Okay, so magnification can be up to 2 million times. Okay, so isn't this wonderful? It's 2 million times. They can magnify it up to 2 million times, whereas a light microscope can magnify up to 2000 times only. Okay, so this is what the difference between the light microscopes and the electron microscopes is. Okay, it is the most important difference because they can magnify up to 2 million times, which is very, very unique for this particular type of microscopes. So this electron microscope looks like this, where they will have an electron gun, magnetic lenses will be there, sample will be placed, and a screen will be there. Okay, and these are electron beams. Okay, these are highly energetic electron beams which are present. Now, let us talk about the two different types of electron microscopes. One is the transmission electron microscope, and the other which is also known as TEM, okay, TEM, TEM in short, and there is another one which is known as scanning electron microscope, which is also known as SEM, SEM. Okay, so let us talk about transmission electron microscope first. So it's a stream of electrons. Here a stream of electrons are formed. Stream of electrons are formed. Okay, then these electrons are accelerated using a positive electric potential. Okay, that means an electric potential should be there inside the system. Okay, it will not be like normal compound microscopes. Then focused by metallic aperture and electromagnets. Electromagnets will be there inside the system. Then interactions occur inside the irradiated sample, which are detected and transformed into an image. Okay, so where the interactions will occur? Interactions will occur inside the irradiated sample. Okay, inside the irradiated sample, which are detected, and then it will be transformed into an image. Now, what kind of image it will produce? It will produce a 2D image. Okay, it will produce a 2D image of an object. Okay, and the magnification, if you see, the magnification is 10,000x to 100,000x. Okay, so can you differentiate between an electron microscope and a normal light microscope? It is having a high magnification of 10,000x to 100,000x. Okay, that means 1 million times. Now, let us talk about the next microscope, which is the scanning electron microscope. Okay. Yeah. So, what happens here in case of scanning electron microscope? I will tell you the difference in our next, slide. So, it scans a gold-plated specimen to give a 3D view of the surface of an object, which is black and white. Okay, so mostly black and white images will be produced, and it will be a 3D view. But what we have discussed in the previous microscope, there that means the transmission electron microscope will provide you a 2D image, whereas here it will provide you a 3D view of the surface of an object. Now, why surface of an object? Why surface of an object? Because see, the specimen that will be there, it will be attached with a gold plate. Okay, a gold coating will be made onto the specimen, and that is why it will means when the electrons will strike on them, they will not be able to penetrate inside the sample. Okay, so what will happen, that they will provide the surface topography or the surface image of the object, which will look black and white. Okay, so used to study surface features of the cells and viruses. So any kind of cell, viruses, or different other objects, this is not only applicable for microbiology, it can be applicable for different other fields like chemistry. It can be, you can see the nanoparticles also, you can see different kind of other objects which are used in physics and chemistry. Then these scanning electron microscopes has a resolution of 1000 times better than a light microscope. Okay, so it is far, far better, but the thing is, this particular instrument is very, very costly, but as at the same time they also requires a specific hand or specific person to operate that particular microscope. Now see here, there are different kind of images. Okay, now let us start with the first tree. Now here you can see this is an image, this is also an image, and this is also an image. These are three different images of bacterial cells. Okay, now what can you differentiate between these three images and these three images? What happens here in this case is that this is a surface topography. Okay, surface topography means it is showing you, or it is providing you a surface image of an object. So what is the object here? Here the object is a bacteria. Now this bacteria means the cell of the bacteria is used to observe the image. So in that case they are providing only the surface structure of the bacteria, whereas in case of this one they are providing means they are providing an internal. Okay, internal structures can also be visualized. If you zoom it further, then you will also be able to visualize the internal structures. So this image is a TAM image, whereas this one is a SAM image. Okay, this one is a TAM image and this one is again a SAM image. Now here, what happened here? Can you recognize this particular image? Here, actually during the preparation of the sample, okay, during the preparation of the sample, this particular image has got disintegrated, or you can say it has got degenerated. Okay, due to handling error. It may happen due to handling error, where you can see that this particular structure is not quite good as compared to that of this D and E image. Okay, so handling of the instrument is very much important. Handling of the samples while preparation, that is also important, because if you cannot handle it properly, or if you don't if you cannot make it while preparing the sample, if you are not concentrating well and you are not able to prepare the sample, then also it may create problem in the future, because it will not provide you the exact image that is required by this particular exact image that you are desired of. Okay, so next, let us discuss about the different kind of features and the differences, the major differences between TAM and SAM. Now, first of all, let us see the types of cells that can be visualized. Types of cells visualized. So here, in case of TAM, before that, let us talk about the source of illumination for image formation. Okay, so here, in both the cases, these are electrons. Okay, there is no light involved in both of the microscopes. Then types of cells to be visualized. Here you can see thin sections of specimens. The electron beams pass through the sections and form an image with high magnification and high resolution. So what happens in this case is that the specimens needs to be very thinner. Whatever specimen you are providing, that specimen needs to be very much thinner so that the electron beams can pass through the sections or through the specimen and form an image with high magnification and high resolution. Whereas in case of this one, that means in case of SAM, what happens is the specimen is coated with gold. Okay, here the specimen is coated with gold and electrons are reflected back. Okay, so what happens is that as soon as this strikes on the gold, they will reflect back. So as they reflect back, so they can provide only the surface topography of the specimen. Okay, mind it. So they can provide only the surface topography. Why? Because as the electrons strikes on the coat, okay, or the gold coat, they will reflect back and provide you with the surface topography of the specimen. Then the next difference is regarding the image. Okay, so in this case, TEM will be providing you a 2D image. Okay, that means a 2-dimensional image, whereas in case of the other one, they will provide a 3-dimensional image or the 3D image. Then let us talk about the nature of lenses. So in case of nature of lenses, both of them have the same kind of lenses or similar lenses they are having. One electrostatic lens will be there and with few electromagnetic lenses will be there inside the system. And here also the same thing will happen. One electrostatic lens with few electromagnetic lenses will be there. Okay, so I guess up to here it is clear. Now let us talk about the medium. Medium means what will be there inside the system. Inside the system, the entire medium will be completely vacuum. Okay, it will be completely vacuum. Okay, so in both the cases it will be vacuum. Then let us talk about the specimen mounting now. Now while preparing the specimen, you will have to mount it, right? So in case of compound microscope, as you mount it with DPX and some other thing. So similarly here it will be a little bit different, but yes, here also specimen mounting is required. So here what will happen, mounted on coated or uncoated copper grids. So here small sized, it can be, you can compare it with this size of a button of a shirt, okay, which is made up of copper. So onto those copper grids, this particular specimen will be mounted. And then in case of SAM, mounted on aluminum stubs and are coated in gold. Here what will happen, they will be mounted onto a aluminum stub, and after that only they will be coated with the gold. Okay, remember this. First, what will happen in case of SAM? In case of TEM, there is no such complexities. Here only copper grids. Okay, onto copper grids, the specimen will be plotted as you have prepared, and then it will be incorporated inside the system, and then you can visualize it. But here what will happen? They needs to be mounted on aluminum stubs, and after they are mounted on the aluminum stubs, then you will have to coat it with gold. Okay, there is a specific machine is available along with the SAM system where it will be coated with the gold. Then next feature is the focusing and magnification adjustments. So this is completely electrical. Okay, focusing and magnification adjustment is completely electrical in both the cases using a deflection coil. So they will use a deflection coil inside the system to monitor the focusing, or you can handle the focusing as well as magnification adjustments. Okay, so here I can show you one. Here you can see that a PC system is there along with the system. So here this is the item or the device through which you can monitor the system, or you can focus or magnify, or whatever adjustments you require, you can do it with the help of this one. So lastly, the means for obtaining specimen contrast. So means for obtaining specimen contrast. Now we have already discussed about different kind of variables when we have discussed about microscopy, right? In the very onset or in the very beginning of our first part of the class. So there we have discussed about different kind of variables, and one of them, and the major one of them, is the contrast. Okay. Now how it is done in case of the SEM and TEM? Here electron scattering will be there, and in both the cases electron scattering will be the means, okay, for obtaining specimen contrast. Okay, how much electrons they are scattering and how they are scattering, depending on that only your contrast will be modified accordingly, and finally you will get a very good quality, high resolution image of the object. Okay, so I guess this will help you. This entire session of part 1 and part 2 of the microscopy session will help you understand briefly at least about the different kind of variables of microscopy. Then we have discussed about different kind of light microscopes which are used, although there are a lot of other light microscopes are also available, but I have discussed about the most commonly used microscopes. And then the final one, finally we have discussed today mainly about the fluorescent microscope as well as the different kind of electron microscopes like SEM and TEM, which are used in the which are very commonly used in laboratories, and for gaining different kind of data for research purpose also you will require TEM and SEM images to project a very good quality image in your manuscript or in your PhD thesis or MSc thesis, or even different kind of project reports you can utilize it. And I hope this particular session will help you in your near future, and all the best. We will be discussing about different other topics in our next coming sessions. Thank you so much. Thank you.